Okay, welcome to the Anatis Knowledge Academy webinar series. Today, we are happy to have Marie-Andre with us to discuss methods for controlling the striped cucumber beetle in greenhouses. Marie-Andre completed her bachelor's degree in biological and ecological sciences at the University of Montreal in 2023. She is now doing her master's degree in plant biology. Her research focuses on finding new ways to control the striped cucumber beetle in greenhouses. So thank you, Marie, for accepting our invitation to share the result of your research with our attendees.
Well, thank you, Omid, for the invitation. So today I'll present to you my project, which is called Alternative Method, Control Method Against the Striped Cucumber Beetle in Cucumber Greenhouses.
Okay, perfect. So before starting, I would like to add that we will also have a short presentation from Vincent today. Vincent is the scientific director of Anatis Bioprotection in division of the biopesticides. So after your presentation, Vincent also add some information and updates for controlling of this striped cucumber beetle. So, and also please, if anyone has any question, just write down, and after finishing the presentation, we will discuss on that. Okay, thank you. So go ahead, Maria. Thank you so much.
Thank you. So I will start with a quick overview of the economic importance of the greenhouse production. So first, in Canada, it represents $2.4 billion in farm gate values, $1.6 billion in exportations, and by itself, cucumber represents 36% of the national greenhouse vegetable production. In Quebec, greenhouse production also represents a significant amount in farm gate value and in exportations. Cucumber represents a third of the provincial production. Although both consumption and production are increasing in Quebec, production does not meet demand. There are various factors limiting production, such as bacterial wilt and its vector, the striped cucumber beetle. So bacterial wilt is caused by the bacteria Erwinia tracheiphila. Its transmission is based on its vector, the striped cucumber beetle, which I will refer to as SCB. And in Quebec, 70% of the greenhouse screened in the monitoring network considered SCB to be a major phytosanitary issue. In organic fields, a few control methods exist. In cultural control, the use of floating row covers, insect exclusion netting, plastic mulches, winter rye cover, and perimeter trap cropping with preferred high cucurbit and squash cultivars have been assessed in several studies. In biological control, 2 parasitoids have been observed in the province. Also, the use of entomopathogenic nematodes shows interesting results. In chemical control, only 2 products are available in organic production. Finally, in semiochemical control, the aggregation pheromone of the striped cucumber beetle is known but is patented. And the use of pheromones for monitoring or mass trapping the striped cucumber beetle also shows promising results. In greenhouses, the only control methods available consist of manually removing the individuals or adding nets to the greenhouse openings. There is no biopesticide available. These methods are laborious and ineffective, so new control methods against the striped cucumber beetle must be developed. The aim of this project is to identify and evaluate repellent molecules in order to develop an alternative control method using a push-pull strategy against the SCB in cucumber greenhouses. The first part of the project is to evaluate the repellent effect of surround WP, or kaolin clay, on the SCB in experimental greenhouses. We want to confirm the reported effects of kaolin on reducing mortality, defoliation, and the incidence of bacterial wilt. The second part of the project is divided in two. First, we seek to identify and evaluate the repellent effect of essential oils on the SCB in olfactometry. Then, among the essential oils tested, we want to evaluate and confirm the repellent effect of rosemary and thyme on SCB in experimental greenhouses. We will also test aromatic plants by itself. Trials about kaolin clay were conducted last summer. We aim to evaluate the repellent effect of kaolin clay on SCB in experimental greenhouses. We hypothesized that the proportion of defoliation would be lower in treated cages than control cages, and that the proportion of dead SCB after 6 weeks would be higher in treated cages than in control cages. The bioassays were conducted simultaneously in 24 muslin cages installed in a complete randomized block design comprising 12 repetitions of each treatment, namely the control treatment, which is water, and the experimental treatment, the surround WP or kaolin clay. Each cage contained 3 tutored cucumber plants at the 3 leaf stages. Kaolin was applied using a handheld sprayer according to the supplier's label. First 2 applications were applied 3 days apart prior to the introduction of the SCB. Subsequent applications were made at 7 days intervals for a maximum of 5 applications. The control treatment was applied at the same intervals and at its own handheld sprayer to avoid cross-contamination. The SCBs were introduced in the cages after the first 2 applications. Since the intervention threshold in the field is 0.5 individuals per plant, and we wanted to observe significant impacts on SCB, 8 adult individuals were introduced per cage. The parameters assessed weekly for 6 weeks were mortality, the presence of bacterial wilt, and the percentage of defoliation for 5 random leaves per plant. To assess the effect of treatment on the proportion of defoliation, a binomial generalized linear mix model was applied using the percentage of leaf defoliation as the response variable. The applied treatment was used as a fixed effect. Random effects included the date of the data collection, the randomized blocks, and the cucumber plants. Then to assess the effect of treatment on SCB's mortality, the same model was applied using the proportion of dead SCBs at the end of the bioassays as the response variable. The treatment was used as a fixed effect. Randomized blocks were considered as random effects. Results show that the proportion of defoliation is significantly higher in control cages than in treated cages, reaching less than 1% per leaf. And concerning mortality, results show that the probability of death after 6 weeks is higher in the cages treated with kaolin clay than in the control cages. Probability of death reaches 28% in control cages and 52% in treated cages. We also observed that the total number of individuals after 6 weeks increased in 2 control cages, which are the only cages where reproduction occurred. As reported in several studies, surround WP reduces SCB abundance and defoliation. However, the experimental treatment did not influence the presence of bacterial wilt. In brief, surround or kaolin clay is a potential alternative to insecticides for protecting cucumber plants in organic management. Concerning olfactometry, trials were also conducted last summer. We aim to identify and evaluate the repellent effect of essential oils against the SCB among the followings. So lemongrass, eucalyptus, wintergreen, rosemary, and thyme. These essential oils showed interesting results in fields and greenhouses against various pests. We hypothesized that every essential oil would be chosen in less than 50% of trials, regardless of treatments and individual sex. We also hypothesized that rosemary and thyme from the Lamiaceae family would have a lower probability of being chosen than the other EO tested. And yes, I will refer to essential oils as EO. So what is olfactometry? Well, it is a system in which incoming air, I'll try to point it to you. So yeah, it's a system in which incoming air is propelled and divided between two branches. The air is first filtered through activated charcoal, then humidified and impregnated with the substance to be tested. The stem of the white tube is connected to a modified valve that allows the introduction of the striped cucumber beetle. The olfactometer is placed under artificial lighting in a black geotextile enclosure to prevent the insect from being stimulated by the external environment. During an assay, the third vial of each sequence contains of a cotton round moistened with 50 microliter of the essential oil to be tested with or without a cucumber plant, and the other one contains the alternative. During an assay, the striped cucumber beetle is released in the system, and if after 5 minutes the individual did not come out of the valve, the test is terminated and then restarted with a new individual. Otherwise, data collection begins as soon as the SCB is released from the valve, and the test ends when the 10 minutes allowed have elapsed or when the individual has made a choice between the two branches here. All EOs were tested in 4 scenarios. First, they were tested alone against an airflow to assess the repellent effect. Then they were tested alone against a cucumber plant to confirm that the EOs are less attractive than the cucumber plant. Then they were tested in combination with a cucumber plant against an airflow to evaluate if the essential oils would be repulsive enough to cover the smell of cucumber plant. And finally, they were tested in combination with a cucumber plant against a cucumber plant to evaluate the capacity of the SCB to distinguish and choose the right branch. For each combination, 24 SCBs were tested, and each individual was used only once in the assays. To assess the probability of choosing the essential oil under test, a binomial generalized linear mixed model was applied using the final choice as the response variable. The EOs, their combinations, so with or without cucumber, the alternatives, so airflow or cucumber, the sex of the SCBs tested, and the position of the white tube were used as fixed effects without interaction, and the date and time specific to each assay were considered as random effects. In the first scenario, where essential oils were presented alone against an airflow, all EOs showed a probability of choice under 50%. Eucalyptus, rosemary, and thyme have significantly lower probabilities to be chosen than wintergreen and lemongrass. In the second scenario, where essential oils were presented alone against a cucumber plant, eucalyptus, rosemary, and thyme showed the probability of choice under 50% and are significantly less chosen than wintergreen and lemongrass. Moreover, lemongrass exceeds 50%, and these results show that lemongrass and wintergreen probably have no effect on the SCB. In the third scenario, where EOs were presented in combination with a cucumber plant against an airflow, Results show that all EOs have a probability of choice lower than 50%. Again, eucalyptus, rosemary, and thyme's probabilities are significantly lower than for wintergreen and lemongrass. In the last scenario, where EOs are presented in combination with a cucumber plant against a cucumber plant, which is similar to a greenhouse context, all EOs show the probability of choice under 50%. Again, these probabilities are a little bit higher than in the third scenario. Different factors can influence the effect of essential oils on the striped cucumber beetle's response, such as the application method. I used passive diffusion, but some studies directly sprays the essential oils on crops. The concentration is also a factor. I used a fixed concentration during the trials, but some essential oils, such as eucalyptus, show the repellent effect at certain doses and an attractive effect at others. The chemical composition of essential oils is also a factor. For example, thyme is known to be more repulsive if it contains more thymol. And finally, concerning the form, there's a study that showed that for a same species, the use of the aromatic plant itself could have a bigger repellent effect than the use of the essential oil, which is the case, among others, for rosemary. Also, remember, there's a difference between when essential oils are presented against an airflow or cucumber. Well, these results led me to question the emissions of cucurbitacin, which is the volatile compounds that attracts SCBs. Well, cucurbitacin is synthesized in response to various factors such as environmental stress, herbivory, temperatures, light, nitrogen supply, and water stress. It is also interesting to mention that cucumber is not SCB's favorite cucurbits. Finally, other factors could influence SCB's response, such as their diet. SCB were fed with cucumber, but were captured in various cucurbits fields. Also, statistical analysis revealed that test time could explain up to 9% of the variation. Concerning the repellent effect of rosemary and thyme, trials will be conducted next summer. Even if eucalyptus showed similar results to those two, we decided to continue with rosemary and thyme because of their repellent effect often documented in the literature and because of their availability. The objective is to evaluate the repellent effect of rosemary and thyme aromatic plants and essential oils on the SCB in experimental greenhouses. We hypothesize that the percentage of repulsivity will be superior to 0 for every aromatic plant and EO tested. We also think that the proportion of emerging SCBs after 7 weeks will be lower in treated cages than control cages. Concerning rosemary, we hypothesize that the repulsivity will be higher and the emergence will be lower for the aromatic plant than for the essential oils. And for thyme, we believe that the effect will be reversed. The bioassays will be conducted in arenas consisting of 2 muslin cages connected symmetrically by a plastic tube. These arenas will be installed in experimental greenhouses. Each cage will contain three tutored cucumber plants at a 3-leaf stage. The essential oils will be tested separately. Rosemary and thyme will be tested separately in a complete randomized design consisting of 2 treatments, so the essential oil or the plant itself, and plus the control, using 10 replicates. During the assays, the tested EO will be passively diffused, or the plant the aromatic plant will be placed in 1 cage per arena. The control treatment will be diffused simultaneously in the remaining cage. The diffusion will start 24 hours prior to the introduction of 20 SCBs in the central tube. The measured parameters will be the number of striped cucumber beetles per cage every 2 days for 6 days to evaluate their repulsivity on time. After this period, all SCBs will be withdrawn of the cages and the cucumber plants will be kept for 6 weeks, after which the number of striped cucumber beetle per cage will be measured to evaluate the emergence of adults. Here's my provisory schedule on 12 weeks. So for the first 3 weeks, I will be preparing the greenhouses and the seedlings. Trials for each aromatic plant and essential oil will take place from week 4 to 6. Week 7 to 9 will be used for redaction and data analysis. And finally, from week 10 to 12, I will collect the data concerning the emergence of adult striped cucumber beetle. To assess the repulsivity of each treatment, a binomial generalized linear mixed model will be applied using the percentage of repulsivity as the response variable. The applied treatment and the time of the data collection will be used as fixed effects. Random effects will include randomized blocks and the week of the data collection. To assess the effect of treatment on SCB's emergence, the same model will be applied using the percentage of emergence as the response variable. The treatment will be used as fixed effect. Random effects will include randomized blocks and the week. I still have a few interrogations concerning these trials. First, concerning the diffusion, I'm pretty sure I'll use passive diffusion because it is, well, cheaper and easier to use. And during the olfactometry assay, I used the filter paper as a diffusion medium. So I was thinking of using the same medium. But some studies use test tube in which they put the right concentration of essential oils to diffuse. So I'm not quite settled on the diffusion medium. About the aromatic plant, I'm not sure yet about the growth stage of the plants that I'll use or how many will be used per cage. I was thinking of one plant, aromatic plant per cage, but it might be difficult to use the same ratio than in commercial greenhouses. Also, to limit the cross-contamination, I plan to use 2 separate greenhouses in which I want to install a curtain to isolate the experimental cages from the control cages during the trials. At the end of this project, there will still be some challenges to overcome. It will be interesting to evaluate and identify the optimal concentration of essential oils and the optimal diffusion methods in commercial greenhouses. It would also be interesting to evaluate the impact of EOs on beneficial insects. One of the biggest challenge would be the registration of essential oils and surround WP for agricultural use in greenhouses. Hopefully, these repulsive molecules will be used as part of a push-pull strategy against the striped cucumber beetles in Quebec greenhouses. Thank you for having me and allowing me to share the results of my project. I spoke really fast, I think. Now I'll do my best to answer your questions because as you've heard, my English is a bit rusty, and I'm also happy to receive your suggestions and comments. Thank you. We can't hear you.
Sorry, my mic was okay. Now can you hear me, Mari?
Yes.
Okay, okay. Thank you so much for the presentation. It was great. And before starting the questions, so I can see that Vincent is also here with us. As I explained, he is a scientific director of Anatis Bioprotection in Division of Biopesticides. So he will also give us some updates on effect of bioseries on for controlling the stripe cucumber beetle. So Vincent, we are with you. That can you also give us some updates and information about the bioseries? Buvaria bassiana. So we couldn't hear you.
Oh, can you hear me?
Yeah, yeah. Now it's okay.
Okay, okay, good. Great. Can I share? I prepared a small PowerPoint presentation as well.
Yeah,
So
perfect. I agree. Thank you so much.
thanks, Marie-André, for this beautiful presentation. Yeah, when I heard that, first of all, yeah, my name is Vincent. I'm the scientific director of the biopesticide division at Anatis Bioprotection. That means that I'm in charge of research and development and quality control, and also involved in regulatory affairs. So wearing all kind of different hats in the company. And when I learned that Omid was organizing this meeting about the strip cucumber beetle, I thought that it would be a good idea to give you a quick update about the trial that occurred in 2023 on this pest. So I just have a couple of quick slides for introductions. I'm assuming, I don't know, does everyone knows that Bioceres is a a Bovaria bassiana based product with the strain ANTO3. We are located in Laval and near Montreal. We have regulation in place in Canada, USA, soon in Brazil too. Um, so I just don't want to go into much details, but so Bovaria bassiana is a entomopathogenic fungus that is occurring worldwide. By meaning worldwide is that there is different strains. You can. I'm quite sure you can take a soil sample anywhere in the world, and you are quite almost 100% sure to find Bovaria bassiana spores. Of course, you may not find the ANTO3 strain, which is the one that we manufacture and sell at Anatis in our products. Our strain was isolated in North America on Lygus bug, tarnish plant bug, actually, near Montreal. So Bovaria bassiana-based products are compatible with most IPM programs. There is no non-resistance mechanism, which is a good idea to use this kind of microbials into conventional rotation program as well to use it as a kind of resistance management tool. The way it works, Bovaria bassiana, is that the product contains the live spores of the fungus. The spores needs to get into contact with the insects, and they will germinate and build a germ tube that will penetrate into the insect's body. And once it's insect the insect's body, uh, the hemolymph is like a liquid fermentation broth, for the fungus. So once inside the insects, the fungus will, replicates with blastospores that we call, and it will invade all the insect's body, produce different kind of metabolites until it kills the insects. It can take up to 4 or 5 days, up maybe up to 7. It depends of the temperature, depends of the size of the insects, depends of the amount of any inoculum that got in touch with the insects. So the speed of action is not it's not a fast knockdown effect like most chemicals, but it's quite effective when you use it under with the right condition. So our products are mostly effective on aphid, thrips, and whiteflies. Those are the big trees for which we have the most studies being conducted. But we also have proof of efficacy towards other pests, such as different beetle species, plant bugs, root aphids, also spider mites, chinch bugs, many more. So in Canada, we have the wettable powder formulation by Fabricius GWP, which is registered for reduction in number of whiteflies, aphids, thrips, and Colorado potato beetles, and also rice root aphids. But this product is targeted for use in greenhouse crops, ornamentals, cannabis, vegetables, strawberries, and herbs and spice. You can use it as a foliar application, cutting dips also in ornamentals, and also drench application for use in cannabis. There is also a emulsifiable concentrate formulation, which is an oil-based formulation, which has about the same use patterns, except that this product is also can also provide a good level of control on mites population. So that was just a big introduction, but the main thing that I want to show you today is just the results of a study that we conducted with a partner based in Ontario in 2023 on strip cucumber beetles, because in the past, Sylvia Todorova, our founder, she did a lot of work on the cucumber beetle with the ANTO3 strain. But most of the studies were lab studies only, and we found that this species, this is an insect species, is the Bovarea bassiana Bovarea bassiana ANTO3 is able to infect the strip cucumber beetle. There is no, it's really, it works very well under laboratory condition. It's just that every time that we sponsored a field trial or a greenhouse trial, there was always some kind of problem occurring, or maybe sometimes it was the experimental design, or we don't know exactly. The thing also is that we found out that the overwintering adults, I mean the ones that are emerging from the field margins in the spring after spending the winter in the ground, those were more susceptible to the fungus. And yeah, so back to the study we conducted last year. So we found that the product used was BioTitan. It's the exact same product. It's just that BioTitan is the trademark for field use, outdoor use. The product was applied at 2 different rates, 2 kilos per hectare and per hectares, and 4 kilos per hectare, with a 500 liter per hectare spray rate. And we found out that we had already after the first the red arrows indicate the date of the application. So, and the assessments were being conducted right before the next application. So we found out that right after the first application, there was already a significant decrease in the number of the percentage of plants being damaged by cucumber beetles. Those were cantaloupe plants between 3 and 6 leaf stage. And after the second application and for the rest of the studies, we recorded a really significant decrease in plants being damaged by the strip cucumber beetles, up to near 90% control. So I wanted to show this today only to let you know that, of course, this use is not registered at the moment. So this is not a recommendation. This is simply to inform you. And at Anatis, we will take the necessary action, to ensure that this use both for field-grown crops and also indoor crop will be allowed, for this product to be allowed for both scenarios for use on the strip cucumber beetles. And of course, we will conduct more studies to confirm those observations. So yeah,
Okay.
that's it for.
Thank you so much, Vincent. But my question is about the registration. Do you have any estimation time that we will have it registered in the market or not? When we will have it to use?
We can't, to be honest, short answer is anywhere between 18 months and 2 years. I'm afraid it's not I would like to be optimistic, but the reality is that we already have a submission under review at PMRA, and we cannot submit another one before this one is really approved. They call it the tailgating policy. So
Yeah, got it. But
yeah.
according to results which I saw and you showed us, it's a very good result. Actually, it's controlled very well.
Yeah, it's really promising. Of course, we will repeat just to ensure that we define the right rates also and use pattern. But yeah, that's really interesting.
And is there any condition which necessary to have, for example, humidity or any temperature which you maybe did or you maybe recommend for using?
In general, we recommend. The optimal temperature for the Beauveria bassiana to be active is between 20 and 30 degrees Celsius. Okay, so above 30, you can apply with temperature above 30. It's just that above 30, there is limited growth of the fungus. You will not kill the spores if you apply at higher temperature. It's just that the spores will remain inactive on the foliage or on the insects. So, but once the temperature cools down, during the evening or during the night, the speed of growth, will increase. So there is no. The biggest concern about applying Beauveria bassiana based product is the UV light, because the spores are killed really fast with the UV radiation. So that's why the main recommendation is to apply during a cloudy day or during late afternoon, early evening. And this is what happened in the study that I've just showed you. All the sprays were applied around 6 p.m.
Perfect,
So I think that would be the best thing to recommend.
And in optimum condition, how long will it take to affect? In optimum condition?
But anywhere between 3 and 7 days.
7 days. And so you also mentioned that below the 20 temperature we can also use, but it will take a time, yeah?
Yeah, it will take more time. Yeah, because as you know, the insects are a cold body.
Yeah.
So the speed of action depends of the insect's temperature, which is related by the air temperature. So it's yeah.
Yeah. Okay. Great. Thank you so much for the presentation and the information and updates. So, if is there any question about both presentations, we can answer or discuss about them. So I'm checking the chat box. Here I couldn't see any presentation. So if there is no question, so we look forward to seeing you all in next our webinars series. So stay safe, and I'm saying goodbye.
Bye. Thanks, Omid, for the
Bye. Thank you.
invitation. Bye.
Thank you.
Thank
Bye.
you. Bye bye. Bye.